Hello, everybody. In today's video, I am planning on covering some mathematical details for fine mapping. You're not required to know this for the subsequent videos, but I do recommend you listening to this video and try to understand because I feel like if I understand the mathematics behind some methods, I can use it better and I can interpret it better. But again, no pressure in terms of understanding everything in this video. So let me start somewhere that you may already be familiar with. And this is the mathematical motivation for phi mapping. And it has to do with linear regression. So we're looking at two types of linear regressions. One is called simple linear regression. And the model is y equals to beta x plus some error term. And it only has one single explanatory variable, or sometimes called the predictor, independent variable, and it's represented by this x. And this is the basic model for GWAS. So in our case, the Y would be some phenotype that you're interested in, and this X would be the measurement of one SNP. And the other one is called multiple linear regression. And from the name, it's obvious it includes not just one, but multiple explanatory variables, and they're all different. So in our case, it could be k different SNP measurements. It doesn't have to be SNPs though, it can be structural variants. I'm just using SNP here for simplicity. Well, so these two linear regressions can behave pretty differently. And I have a toy example here. It just shows you how they can produce different results. So suppose you're interested in LDL cholesterol level and you have collected all these data from different individuals and you've measured two SNP locations, SNP1 and SNP2, and all the LDLC measurements for all these individuals. What you don't know is that this unobserved ground truth that SNP1 is causal for the LDLC levels, but SNP2 is not. So in fact, LDLC level is entirely determined by SNP1 with an effect size of 20, and SNP2 contributes 0. But the funny thing is that SNP1 and SNP2 has correlation 0.795, which is pretty high in terms of correlations. So we perform simple linear regression where we regress on one SNP at a time. So if we do this on SNP1, then we get the estimated effect size to be 20, which is exactly the truth. But then if we regress y on SNP2, we still get a pretty high number, 15.9. And this is elevated purely due to the fact that SNP2 is highly correlated with SNP1. So let's see what happens if we perform multiple linear regression. So if we do that, then we can see that the model puts both SNP1 and SNP2 into consideration. And now the estimated effect size is 20 for SNP1 and 0 for SNP2, which is exactly the ground truth. So we've recovered the ground truth accurately with multiple linear regression. And this is because by including the causal SNP in the model, the non-causal SNP no longer has elevated effect size estimate because it doesn't have to explain any signal. So GWAS is essentially built on simple linear regressions, and it tests one SNP at a time against the phenotype. So I have to admit that when I first started learning the word GWAS, genome-wide association studies, I thought that they must be modeling the entire genome. Otherwise, why would they call it genome-wide? It was not until later, took embarrassingly long time, that I learned the word genome-wide is referring to the scanning range. And the test itself is only performed one variant at a time. So I was a little disappointed. But then I learned that people didn't stop at simple linear regression because a lot can be gained by going beyond association and try to get to causation. And this is what fine mapping is about. We're not quite modeling the entire genomes yet with fine mapping, um, but we do model one region at a time, and it's a pretty big region, usually 3 megabase pairs. And it does take all the variants in that region into consideration. you may have questions, why 3 megabase regions and how exactly do the model take all the variant into consideration? So that takes us to the next section. These are great questions, by the way, that I came up with that I thought that you would ask. So the how of defining these 3 megabase pair regions is that we iteratively find 3 megabase windows around lead SNPs, and I'll talk about this a bit more in a few minutes. But the goal, though, is that we want to separate out LD independent regions. And in other words, that we want variance in different regions to not be correlated. And let me show you why we want LD independence and how it can help us. So this is another toy example. Still LDL cholesterol levels. So it's the same phenotype from our previous toy example. Now we're in an alternate universe where 2 out of the 3 SNPs you have measured contribute to LDLC. So the SNP1 contribute 20, SNP2 contribute 10, SNP3 contribute 0. You still don't know this ground truth though, but you're trying to find it out. And in this universe, you're in luck because none of these SNPs are correlated with each other. And you can go ahead and do a simple linear regression. One SNP at a time, you find the effect sizes, and it's exactly right, 20, 10, and 0. And you just want to double check with multiple linear regression. So you do multiple linear regression, and the answer come out 20, 10, and 0. So what we have seen here is that when things are not correlated, then we don't have to look at them together. We can divide and conquer. So that's the reason behind why we want LD independence. But why 3 megabase pair regions? And the answer is in the human genome across many ancestries, LD decays pretty quickly after a few kilobases. 3 megabase pair is just to be safe, and many fine mapping analysts use 1 megabase regions. Here's a plot from the very famous 1,000 Genome Project, phase 3, that a bunch of very hardworking scientists had went and gathered genotyping data from many different people. They had plotted the LD decay. As you can see, after a few kilobases, not even megabase pairs, the LDs start to be pretty minimal, below 0.05. So then now we have established that 3 megabase pair regions around lead SNPs is a pretty good way to get fine mapping started. Then how exactly do we divide up the genome into these 3 megabase pair regions? And the answer is iteratively. So I have kind of drawn this toy example of the genome, and this is the GWAS significance threshold. Y-axis is the negative log 10 p-values, and x-axis is the genomic positions of the variants. Each dot is a variant. And this is how big a 3 megabase pair window is. Of course, the genome actually is much bigger than this, but this is just a toy example. All right. So in the first iteration, as you can see, this one is the highest peak in this genome. So in the first iteration, that's what we find. After we find it, we would cross it out. So we no longer consider this region. And the second region that we find, it must be here. So we find this 3 megabase pair window during the second iteration. And here's the third one. We cross it out. No longer consider any of the variants in the subsequent iterations. And then we have the fourth. You get the idea. And forgot if this is the fifth or, yeah, fifth. And then the sixth. All right, so now we have hit our end condition where nothing in the genome that's left are genome-wise significant. Then what do we do? Sometimes there's an optional step that the analysts would merge overlapping regions. because overlapping could potentially mean non-trivial correlation between the regions, and we don't want that. So if things have notable correlation, the most rigorous thing to do is to put them all together in the model. So if you merge it, then now we have 4 total regions to consider. But in practice, due to computational limitations, people would often choose not to merge the regions, and that's okay too. All right, so now that we have our regions, each of these regions contain tens of thousands of variants. So how do we model them all? Good question. And this is one of my first questions after learning about fine mapping. So I asked, would just multiple linear regression work? If GWAS test 1, fine mapping can just test the multiple. Well, I was a bit naive back then. If I had given it more than 5 minutes of thought, I would have known that this is not going to work that well. So the answer is not a straightforward multiple linear regression. And why is that? There are 2 related problems. The first problem is the m greater than n problem, where if we regress y against x and x is a matrix that's wider than it is tall, then this system of linear equations that we set up will not have a unique solution, and the math will just fail. However, you might say that, okay, in larger biobanks, we have more sample sizes. This will not be a problem because typically we have more than 100,000 individuals in a large biobank, and each region typically contains about 20,000 variants. So this matrix will still be taller than its wide. So even if we can solve this m greater than n problem, there's still another problem, which is the highly correlated variant problem. And this is very common in real data, and it's not going to get resolved by increasing sample size. So I want to show you what happens when there's highly correlated variance in a region. So again, let's see this toy example of 5 highly correlated variants in a region, and only one of them is causal, like this third one has non-zero contribution to the phenotype. But again, you don't know this. This is just unobserved ground truth. But what you can know is that after you measure all the SNPs, you can test their pairwise correlation. And it happens to be 0.99, which is pretty high. But in real data, this is actually not uncommon. Let's see what happens. In GWAS, you can probably guess that all of them will have pretty high significance because they're highly correlated to the causal variant, and there is actually a causal signal in this region, so all of them are above significance threshold. But what happens if you want to try and isolate the causal variant using multiple linear regression? So you can definitely perform a multiple linear regression, and it will give you effect size estimates, but you will also see that the effect size estimate is highly unstable because it has really big error bars. The standard errors are too high. And what this would result in is that if you try to compute the p-values for all of these estimates, none of them even would pass nominal significance of 0.05. But being a good scientist and being hopeful, you might say, well, this is salvageable because I know there's a signal here, and GWAS results show you that there's highly significant. And if I were to guess based on the relative significance of all these points, I would guess that the variant 3 is the driver of the signals in the region, also known as the causal one. It looks like I'm writing poetry, but I'm actually not. I'm just trying to set up for a big reveal here. So you're right, this is salvageable. And you're right to guess the top point. So what you have constructed here are prior belief This is you believing that there's a signal here, and you have good evidence to believe that. And also, you have included the data, the relative significance. The GWAS gives you this data. And you have thought about the model. You must look at all the variants in the region, and the model need to include all of them. And finally, you're guessing, which is good, but more rigorously, we would call this inference. So what we have constructed here are basically all the ingredients for Bayesian fine mapping. All right, so now we're ready to talk about what exactly is in the black box of fine mapping. And many people do consider fine mapping a black box, but today I want to show you that it's actually not that complicated to understand it. We're going to focus on this simple method called the approximate Bayes factor method. We call it ABF for short. For people who are a little bit familiar with the field of fine mapping, you might see ABF and you might think that, this is an old method. It was published in 2007. And in this rapidly developing field of fine mapping and genetics research, 20 years seems like a long time. But ABF is actually a great method to start with when you want to study fine mapping. First, because it's relatively easy. The math is simple to understand. And more importantly, because it forms the foundation of pretty much all the modern popular Bayesian fine mapping methods. You may have heard of FineMap and SUSY. These methods are basically built on ABF. And understanding ABF will give you a solid foundation of understanding these more complicated methods if you need to study them in the future. So first of all, let's talk about what the inference target is. And it's this posterior probability P of MC given data. MC is the causal configuration. So what is a causal configuration? For example, if you want to compute the posterior probability of the second variant in the region being causal, you would want to compute the quantity P of M2 given the data, where M2 is a causal configuration indicating the second variant is causal. So only the indicator 1 is at the second position and 0 everywhere else. And the causal configuration is a vector of length m being the number of variants in the region. All right, so we want to get at this posterior probability. And how we want to do it is through Bayes factor. And I'll go through the derivation of the main equation here. So again, this posterior probability, but we want to write it in terms of likelihood and priors. So this step is just using the Bayes rule. And you rewrite it as the probability of data given the causal configuration times the prior for the causal configuration divided by the probability of the data. So now we want to express the denominator in terms of likelihoods as well. And this step, we use the law of total probability. So now we write the probability of data in terms of the sum of probability of data given a causal configuration times the prior for that causal configuration and summing over all the possible causal configurations for this region. And the next is pretty simple. We just divide the top and bottom by the likelihood of the data given the null model. So M0 here represent the null model, and it means there's no variant in the region is causal. So why are we doing this? This looks like the equation is getting longer and longer. Is it helping us? Well, yeah, actually, it is, because now we can express this posterior probability in terms of Bayes factors. So the definition of Bayes factor is the ratio of the marginal likelihood of the data conditioned on the alternative model MC and the null model. So basically I'm saying that this fraction here is just a Bayes factor. So I'm rewriting everything in terms of Bayes factor. And you might still ask, why does Bayes factor help us? So there are actually a few really convenient things we have going on with Bayes factor. We use Bayes factor instead of the likelihood because there are a few unimportant constants that cancels out when we compute this ratio. And it's just mathematically cleaner to calculate. And the Bayes factor is actually more interpretable. So if you think about it, a raw likelihood, like P of data given MC, can take the value of, say, 1.5 times 10 to the minus 16. And if I tell you that, you will have no idea if that's a good likelihood or a bad likelihood. But Bayes factor actually lives on a relative scale because it compares with the null model. So if I tell you the Bayes factor of MC against M0 is 10, then you know exactly what I'm talking about. You know that the likelihood conditioned on the alternative model is 10 times bigger than the likelihood that's conditioning on the null model. Okay, so now we've convinced ourselves that Bayes factors are useful for us. Let's see how it's computed. Let's make this simplifying assumption that there is one and only one causal variant in the region. So what that means is, for example, under this assumption, all of the causal configuration in a region with four variants are these four vectors. This will really simplify the way that we compute the posterior probabilities. Here is the formula for computing the Bayes factor. So all of the quantities in this formula can be found in GWAS. The SE of J is the standard error of the estimated effect size beta J hat of variant J. And ZJ is the Z-score, and W is the user-specified prior variance of the true effect size beta, assuming that this variant is causal. And the default setting for W is 0.04. And this number is chosen when the original paper was written. And since then, some researchers have looked into how to change this prior and how robust this prior is. But I think at this point in the field, we don't really have a consensus on what's a better choice than 0.04. So many people are still using this quantity for W, and you can use it too. This is the Bayes factor, but then there's another component to compute the posterior probability, as you can see here, that here's the Bayes factor, but there's also these priors, P of MC, P of MT. So let's think about the choice of causal priors. These causal priors represent your belief of how likely the variant is causal before you have even seen the GWAS data. And most people use what's called the uniform prior, which treats every variant as equal before seeing the GWAS data. But another way is that sometimes you might want to set the higher prior for variants with more active functional role, like coding variants, and lower for the ones that are not obviously functional. These are called the functionally informed priors. So there are some algorithms that will calculate the functionally informed priors for you after you give it functional annotations. So putting the Bayes factors and the causal priors together, the posterior causal probability of a variant J is its Bayes factor normalized by the sum of all the Bayes factors in the region. So it comes down to a pretty simple formula. And this is very easy to calculate. You can run the ABF method for a phenotype on your laptop within seconds. The higher ABFJ is relative to all the ABFI values for all the other variants in the region, the higher its corresponding posterior inclusion probability will be. And that's obvious from this formula. And ABF has this property of being roughly a monotonic transformation of z-scores, meaning that the higher the z-score is for a variant, the higher its ABF is going to be. Therefore, the highest PIP is almost always assigned to the lead variant. There are some exceptions to this, though, but mostly this is the case. But the magnitude of the top PIP depends on the significance of the lead variant relative to the others. So you can have a lead variant that is much, much more significant than all of its neighbors, then it's going to get a top PIP, the PIP for it is going to be 0.9 or 0.99, something like that. Or you can have a region that's got a lot of highly correlated variants, and all the variants are going to share some PIP, You're still going to get the lead variant having the top PIP, but the magnitude of its PIP is probably 0.1 or something like that. All right, so that's basically all I want to say about ABF. But at this point, you might be questioning the assumption of ABF. There are a lot of evidence for multiple causal variant per region, though, especially for common complex traits. So you might be worried that ABF is not getting at a lot of the variants. What if the top variant is a false positive? Those are very legitimate concerns, and researchers have spent years trying to solve those problems. The causal configuration can have more than one non-zero entries. It can be of this form or that form, two causal variants, three causal variant. It's all possible. And I just first want to be clear about the computational burden of modeling multiple causal variants. And then we'll talk about the solutions people use. So looking at this formula again, that in order to compute the posterior probability, you need to divide the base factor by the sum of all the base factors. Problem is that the denominator, the sum of all the base factors, sums across the set of mutually exclusive and collectively exhaustive events. And that's a lot of events if you model multiple causal variant. If a region has 4 variants, this is not realistic because regions are much bigger than this. But let's start here. If we just assume one causal variant, then this set of mutually exclusive and collectively exhaustive events have 4 choose 1, 4 elements. And if you assume 2 causal variants, then this set consists of 4 choose 2, 6 elements. If you allow all possible causal configurations, 1, 2, 3, 4, then the set consists of 2 to the fourth minus 1 equals to 15 elements, minus 1 just excluding the model that has no causal variance. All right, so 15 is manageable, but let's look at a slightly more realistic example. If a region has 1,000 variants, so you can imagine the combinatorial burden that we have when we assume multiple causal variants. What do researchers do to try to solve this problem? So there's mostly two schools of thoughts. One is to reduce the search space. So if we can't exhaustively compute all 8.2 trillion base factors, how about we search for the most important ones to compute and ignore the rest? And that leads to the method of FIMAP and DAPG. So FIMAP searches this space stochastically, meaning randomly. Every time you run FIMAP, it'll give you a slightly different result, but it's been proven to be pretty robust. DAPG is also searching the space, but in a deterministic way. So the other school of thought is to remove the need to consider a massive search space altogether and turn the inference of every causal SNP into a separate problem that essentially goes back to using ABF's single causal variant assumption. This is the philosophy behind the popular phi mapping method, SUSY. So that's all I want to talk about in this video. I have listed here 4 papers that you can read if you want to dig deeper into the methods.